Alright everybody, this time we are going to get into the world of meiosis, and we're going to do this a little bit different than the mitosis one. Instead of going over all the individual components of each stage of the cell division, I'm going to emphasize things slightly differently here. Now, with mitosis we started diploid and we ended diploid. We started with one cell and we divided it into two twin daughter cells. In meiosis, we are going to create gametes, which are sex cells, which would be egg or sperm. And when we are diploid here, each chromosome has a backup. Okay, di is 2, so the same genes on here are the same genes on here, but they might be different alleles. So we could have one that could be dominant and one that's recessive. They could be homozygous, having the same allele on either chromosome, but basically we have backups when it's diploid. Sex cells don't have backups. So when we start here, diploid, we're going to finish haploid. And so we're going to go through two divisions. So in mitosis, we divide it once, we end up with two daughter cells. In meiosis, we divide it once, have two cells, then we divide them again. We end up with 4 cells that could possibly all be slightly different from one another. So what I'm going to emphasize here are the two levels of division. Our finished product is haploid instead of diploid. And then there is also variation that's created through sex. So let's get started. So in the beginning, the interphase of the cell cycle is the same whether it's a body cell or whether it's a sex cell. You're still going to have the growth phase, you're still going to have DNA replication, and all of that's going to be the same. And you're still going to have the G2 phase where it grows slightly more and double checks the DNA for codes. Again, I'm not going to show all of the formation or the condensing of chromosomes and everything like that with this version. But if we imagine this is the S phase of interphase of the cell cycle, we are going to duplicate the DNA, we're going to replicate it, and it's during replication that we create the sister chromatids. What we're seeing here, when I did the mitosis one, we had only one chromosome with a backup. Now we have 2 chromosomes, with each having a backup. So these are going to code for different genes than what these would code for. So, moving right along. So we have just completed the S phase of interphase. We would go through now during the G2 phase, growing further, double checking the DNA for mistakes, fixing the mistakes if it can, and then we're going to go into the first division. And as we go into cell division again, we get rid of our nuclear membrane. We're going to have centrioles moving. We're going to have these things condensing, which I'm not going to quite show. And as we go here, in meiosis, which is different than mitosis, thank you, Michaela, we are going to take these and form tetrads. And tetra means 4 here, and they are still going to line up at a metaphase plate. But in this first division, what we have here is these tetrads forming. So the homologous chromosomes, this is homologous for this because they are the same genes, and they all come together. Instead of in mitosis going like this, we form tetrads for the first division, and these would be all wrapped up, and it's during tetrad formation that we can possibly get some crossing over to occur, where part of one homologous chromosome exchanges with another. So that is also going to add variation. So we have crossing over occurring during tetrad formation. Now again, one of the ways we can have random assortment and have variation as well, they independently assort, is these could line up like this, or they could line up like this. And if they divide like this apart, these two cells would be different than if they divide like this. That is variation, and this is completely random. They randomly assort which is going to be on which side during that first division. But for this sake, we'll leave it, let's say, like that. We had crossing over. They independently assort. Each of these homologous chromosomes independently assort from one another. And then we're going to have our division. So these are going to reach out. Our centrioles are sending out our spindle fibers. Those microtubules, those protein cytoskeleton, are going to grab onto the centromeres. And we are going to, in the first division, divide the homologous chromosomes. We are going to split these homologous chromosomes. So here we go, division number 1. When we do that first division, we still have every gene from the original. Okay, so we have our first division, comes in, we get split, and that is division number 1. In division number 1, we have tetrad formation, we have crossing over, we have independent assortment, all of that wonderful stuff. And now we're going to start into division number 2. And now with division number 2, it's going to look more like meiosis, where we are going to split apart sister chromatids. So now during the second division, again we have our metaphase, and spindle fibers are going to get sent out as well. But it is this second one that is going to turn these cells haploid instead of diploid. Spindle fibers come from the centrioles. They reach out and grab onto the sister chromatids. We're going to have a checkpoint in cell division in meiosis as well to ensure that non-disjunction does not occur. But in this second division, we now pull apart the sister chromatids. And these get pulled apart towards the separate poles, pulling them apart, pulling them apart. And ultimately at this point, we have our second division. And in our second division, these get pinched. And with meiosis, we ended up with 2 identical daughter cells. We end up with 4 haploid, half the number of original chromosomes. We still have all of the genes, but we don't have backup copies now. And that's when fertilization occurs. If we bring together sex cells, an egg and a sperm, and fertilize, now it goes back to diploid. But right now, we still have all of the genes, but we have variation. Notice when you look at these, we started with 1 cell, but we make 4 possibly unique completely unique sex cells. None of these are identical to anything else because of independent assortment and crossing over. And again, our finished product, I just want to emphasize here, we end up with haploid versions and variation because of meiosis, which is all about creating gametes.